How does neuroimaging research account for medication effects when studying schizophrenia? Have you ever wondered how scientists study the brain in people with schizophrenia while considering the effects of medication? This is a fascinating area of research that combines neuroimaging techniques with a deep understanding of schizophrenia and its treatment. Let's break it down. Neuroimaging research must take into account the influence of antipsychotic medications. These medications can change brain structure and function, which might complicate the results of imaging studies. One effective strategy is to study medication-naïve patients. These are individuals who have never received antipsychotic treatment, often those experiencing their first episode of psychosis. By focusing on these patients, researchers can identify brain abnormalities that are directly related to schizophrenia itself, without the interference of medication effects. Another approach is using longitudinal designs. This means researchers conduct neuroimaging studies at multiple points in time. They scan patients before and after they start taking antipsychotic medications. This allows scientists to observe changes in the brain over time and helps differentiate between changes caused by the illness and those induced by treatment. For example, studies have shown that certain brain regions, like the caudate nucleus, may shrink after treatment with specific medications. Researchers also compare patients who respond well to medication with those who do not. This comparison can reveal differences in brain structure and function that may highlight both the underlying disease mechanisms and the effects of medication. Treatment-resistant patients often show more significant reductions in gray matter and altered blood flow in certain brain regions compared to those who respond positively to treatment. It is essential to control for the type and dosage of medication used in studies. Different antipsychotic drugs can have varying effects on the brain. Researchers carefully document the specific medications, dosages, and duration of treatment to adjust their analyses accordingly. This helps ensure that the findings are as accurate as possible. Using multiple imaging techniques is another way to account for medication effects. By combining structural magnetic resonance imaging, functional magnetic resonance imaging, diffusion tensor imaging, and magnetic resonance spectroscopy, researchers can capture a wide range of brain changes. For instance, measuring functional connectivity and white matter integrity can help predict how well a patient will respond to treatment. Antipsychotic medications can also change the way brain circuits function. Neuroimaging research identifies patterns of brain activation and connectivity that shift with treatment. This helps distinguish between temporary changes due to medication and more stable features of the illness itself. Finally, researchers consider other factors that might influence the results. This includes the duration and severity of the illness, along with other clinical variables. By taking these factors into account, researchers can clarify whether the differences observed in neuroimaging studies are due to the pathology of schizophrenia or the effects of treatment. In practical terms, accounting for medication effects in neuroimaging research is vital for improving the accuracy of biomarkers for schizophrenia. This knowledge helps identify which brain changes are intrinsic to the disorder and which are a result of treatment. Ultimately, this research guides better diagnosis, prognosis, and personalized therapies for individuals living with schizophrenia. By carefully designing studies and analyzing data, neuroimaging research continues to advance our understanding of the biological basis of schizophrenia while minimizing the impact of confounding factors.